Okay, hi guys, hello. Yeah, I'm Mr. Khor. Today we're looking at Wilayah Persekutuan Kuala Lumpur 2025 biology paper. Now, if you're looking for trial papers, you can log on to edukaji.my. If you're looking for any SPM biology courses, you can check out spmbiology.com as I'll be starting my new intake soon in December 2025. Of course, not for you students who are sitting for SPM soon, okay? Now, and if you need some motivation, can check out some of the videos I did with some of the top students this year. For 2024, meaning those who took 2024, they just got their results in 2025. Okay, guys. Now, let's start with question one. Let's start with question one. This is from Form 4 Bab 14. Okay, Form 4 Bab 14 actually came out quite thorough in 2024, but doesn't mean it can't repeat. Yeah, this topic they can ask in structure, they can ask in essay. It's a very balanced chapter whereby the probability to me is about the same because why there are so many components in Bab 14. Okay, first of all, label tendon in diagram 1. Guys, there are many parts where you can just label the tendon. Okay, all right, any of this is fine. You don't have to label 3, 1 is enough. Okay, first of all. Number 2, draw one star because when it comes to tendon and ligament, okay, tendon and ligament, they love to ask about characteristic. You can highlight this. They love to ask chiri. Whenever they ask characteristic of tendon, you can say it is strong. I'm giving you extra point. Strong, flexible, and inelastic. But when we talk about ligament, you say that it is strong, it is elastic. So why must it be elastic? Because bones are hard. Bones cannot contract and relax like muscle. So the ligament itself must be elastic. Okay, mesti kenyal. Yeah. Okay, based on diagram 1, explain movement to bend the arm. Be very careful whether they want to bend the arm, to straighten the arm, or both. Okay, first of all, in order for us to bend our arm, the bicep must contract, tricep must relax. It's quite a standard answer. Standard question, standard answer. You can say the pulling force is transferred from the muscles to the bone. And one more point which I normally highly recommend is radius and ulna is pull up. Radius and ulna is pull upwards. Is pull upwards. Okay, radius and ulna is pull upwards. Guys, this is a K-bar but very common K-bar question. If the tendon is torn, the arm cannot bend, he's unable to move his arm. It's a K-bar question, so its meaning is hypothetical. Yeah, he is unable to bend. Yeah, he's unable to bend would be good. Next, question two. This is something that I have been starting to tell students about since last year, since 2024. Guys, Form 4 Chapter 2. Form 4 Chapter 2, Unicellular. Okay, so far they haven't really touched on it in KSSM. Yeah. Name the organism, guys. Don't forget to underline. A lot of students forget to underline. The underlining is more important than the species SP at the back. Now, food particles will enter the oral groove. Okay, for those who do not know, this is called the oral groove. I always use the example of our mouth. Okay, students, from our mouth, food will enter the esophagus. Okay, sometimes in my class, I trick my students. Hey guys, food from the mouth enter the tract. Then some funny fella will say, trachea. Then I'm like, oh, you are very smart. Food enter trachea, tracheal, bronchus, bronchial, alveolus, and then what? Food exchange. Yeah, this is just to remind them that it's a common error among students. So from our mouth, food enter esophagus. From our mouth, food enter esophagus. From the oral groove, makanan akan masuk gullet. This is gullet. And obviously a food vacuole. Guys, obviously a food vacuole is formed. Okay, say the function of M. M is the cilia. M is the cilia. So it is to allow movement to carry out locomotion through rhythmic beating. Okay, to me, this is important because every time you see movement, you will see rhythmic beating. Okay, so don't be surprised if they ask 2 marks. Hey, sir, why 2 marks? Huh? Oh, number one, to allow movement. Number two, rhythmic breathing. Beating. Okay, like pulse like that. It's like pulse like that. Okay, beating. Acid spill threaten the paramecium. How can reproduce? Okay, in favorable states, it will carry out asexual. Not favorable, it will carry out sexual reproduction. That's the only thing that they want to test you here. Okay, and then after placing paramecium into the seawater, because seawater is salty, which means, of course, this is a K-but. How does N change in the water? Okay, it will carry out more osmoregulation. We should say more osmoregulation is carried out. So rate of contraction of contractile vacuole will decrease. Hey, wait, hold on. X is put in I loud. I loud means the seawater. How N change? Oh, okay. How N change? So we can say that, yeah, you can just say that osmoregulation is carried out. Yeah, this is okay. Osmoregulation is carried out. The contraction will be lesser. Okay, the contraction will be lesser because uh the reason is because the salt water is salty. Seawater is salty. Okay. So because outside is salty, right? Um, because outside is salty, we say that it will slow down until it stops. Yeah, the contraction of contractile vacuole will slow down and then it will stop. That's when the paramecium dies. Okay, guys, once we put paramecium in seawater, after a certain period of time, it will die lah. It will die because why? Outside is too hypertonic. This is because outside is too hypertonic. It is unable to withstand. Okay. So this is okay. Osmoregulation is still carried out, but after that, the contractile vacuole will decrease. If you really want to add in one more sentence, you can say this is because seawater is hypertonic. Seawater is hypertonic. Okay. Okay, guys, coming to this is question 3. Question 3 from Form 4, Chapter 4. Form 4, Chapter 4 under nucleic acid. Two examples of nitrogenous base. RNA. Now, I always tell students RNA is naughty. Wow, so what is this? Suddenly, what RNA naughty? RNA naughty, RNA no T. Tak ada T means ATGC tak ada T ini menjadi U. You can use uracil. Okay? You can use uracil or you want to use cytosine. Yeah. Up to you, ATGC. Polynucleotide, guys, the only thing to take note here is because, guys, what's the difference between RNA and DNA? The difference is one is single helix, one is double helix. One is single strand, one is double helix. Sebab ini RNA, kamu hanya lukis satu polynucleotida. You only draw one. You only draw one. And take note, they give you 3 nucleotides only. Now, you may ask, sir, the nitrogenous base, can I draw the same shape? Can also, but it's okay to have different shape. Because why? It represents either A or U or G or C. No problem, even if three have different shapes. Okay, next, two importances of nucleic acid is just taking the point from the textbook to throw in. Yeah, so you can talk about DNA, you can talk about RNA. RNA is involved, it is highly involved in protein synthesis. Personally, this is just my opinion. I really disagree that they took mRNA, tRNA, rRNA, and then they put in Form 4. This one because guys, I've been teaching STPM for more than 10 years. I teach STPM, I teach A levels. So when KSSM came out, I was so excited. I went through the whole thing. There were certain things which I think is way too deep for students. Example, glycolysis. Glycolysis, tRNA, rRNA, mRNA. Do you know in Form 6, STPM, now I can touch on this just 1 minute. Protein synthesis is divided into transcription as well as, yeah, my eye became old. Transcription and also translation, right? Hey, transcription alone, I take 1.5 hours to cover in STPM. There's a lot of basic stuff that we need uh, for transcription. After transcription, then translation. Yeah, this is where all the RNAs are involved. But the thing is, what for bring up if you're not teaching the whole thing, then students will be a bit half hanging. Okay, so for my students, I would recommend that you remember all the three RNA is involved in protein synthesis, number one. Number two, there are two stages in protein synthesis, transcription and translation. So transcription here, right? It is where mRNA is formed. Okay. From DNA become mRNA. Yeah. So that's it lah. That's all you need to know. Okay, next, moving on. Predict the effect of nucleotide sequence change on synthesized protein. When there's a change in the sequence, you can see here. You can see here. Yeah, quite a few changes lah. Huh? So what happens is the protein cannot function. The protein cannot function. Guys, don't talk about mutation. I know some of you put mutation. Mutation is not like that lah. Not like that. Okay, question 4, guys, from 5 chapter 8. Just yesterday I had my class. After finishing my class, I think that class is about 70 students, 60, 70, all habis already. One student came to me, "Sir, how to study Bab 8?" I'm like, how come you ask about Bab 8? Yeah. Rarely students ask me about Bab 8 this year. Rarely. I'm surprised. Okay, look. Oh, sorry, one more sentence. He said that it is his weakest topic. Yep. Okay, hierarchy of this one here. So this is a bacteria, to be exact, eubacteria. Now you can even roughly guess lah, guys. You can roughly guess from here. Yeah, bacilli. Okay. So the kingdom is eubacteria. It is prokaryotic. The shape is spear, it's a spear shape. Spear shape. Okay, state the differences between the organisms. Okay, so protista like amoeba, all those are eukaryote, but this one is prokaryote. Nitrogen cycle. This is a K-BAT question. Predict what will happen to dead animals if X were absent. Now X is uh, bacteria involved in decomposition. So no decomposition can occur. Okay, no decomposition can occur. And then compare Y and Z. Y and Z. Basically, both are nitrogen fixing bacteria. So example Y is probably Rhizobium. Okay, maybe Z is Azotobacter or Nostoc. Okay, they are what you call it? They are of different names, but they play the same role. That is, they fix nitrogen to nitrate. Okay, so they both fix nitrogen to nitrate. Both fix nitrogen to nitrate. Okay, guys, question 5. Guttation, form 5, bab 2, right? Form 5, bab 2. Now, what is P? What is P? Now, P is, yeah, okay, water droplet. Water droplet can. Secretion of water droplet. So guttation, I think this one the answer gave wrongly. Guys, normally, right, when I prepare just so you know, when I prepare this, right, I don't follow the answer too much because sometimes got a bit of mistake here and there. Yeah, this one I think they put what? Don't know what, no stoma or something. Yeah. Because I have to know what the schema put. Reason being, students may ask so that I'm in the know. Oh, that question. Yeah. Oh, Selangor, the labeling was out totally. Okay. Comparing, they draw one star here. They love to compare this. They love to compare these two, transpiration and guttation. Okay. So just so you know, transpiration is during the day, whereby water is lost from the stomata kan. But guttation only occur late at night or early in the morning because because uh they have to excrete the water. They have to excrete the water. Okay, now this is a good K, but I quite like this question. When I saw this, I'm like, wow, this is not bad because near the sea, right, the sunlight is hotter, the light intensity is higher lah. This is a better word. So evaporation is much higher, more water is lost, transpiration rate is higher. Okay. State the effect of water shortage on stomatal openings of the plant. Now, when water is not enough, what happens is it will close the stoma. It will close the stoma to prevent more water loss. Yeah. Oh, sorry, hold on. Water diffuses out of the gut cell. No, no, this is not so good. This is not so good. Okay, this is it reduces water loss through evaporation. Because now memang also not enough water already. Okay. Next, growth cycle. Growth cycle. Okay, if you are SPM 2025 students, I think it's time to take note. Yeah, it's time to take note. There are quite a number of things that are highly anticipated this year. I mean, from what I have researched, one of it is Form 5 Bug 13. Form 5 Bug 13. Okay, Form 5 Bug 13 um haven't asked for the past 2 to 3 years. Yeah. Last time they did ask a little bit lah about genetic engineering. But this year, if you are 2025 student, please take note of Form 5 Bug 13. This is also one chapter, Form 4 Bug 15. Both ending chapters are very important this year. Okay. This is Form 4 Bug 15. Okay, X here is what we call larva stage. And one example other than the grasshopper is Butterfly. 6A1. Yeah. Yeah, correct. Okay. So butterfly. Butterfly. And you should draw something like this, but I have one quirk with what they gave. This is what I followed from the schema, but I think don't have to put so many times. Yeah, because here also they showed a very simple few stages only. So guys, I would say right for this one here, maybe at least three stage should be fine lah. To me, right, three stage. Three instars would be good. Three instars would be good. Please take note of one thing. Instar is the period between ecdysis. Ecdysis. So we do not count the first and last as instar because what is instar? Period between. Between means before, after. Look, here don't have. Here don't have. After God. Before don't have. Then towards the end, the adult stage. After don't have. Before God. Before God. Okay. Okay, this one nothing much, guys. So for 6.1, 6.1 is complete metamorphosis. No acidisis, but 6.2 incomplete. Incomplete. But got acidisis. Ecdysis, ecdysis. Yeah, if you forgot, guys, I'm not too sure what this. Please read up. Please read up. It's already the last day of October. All right. This is a K-bar question. This is a K-bar question. You see, they love to use alphabets to represent the individual. Okay, based on the diagram, explain the effect of insecticide on the life cycle of the insect. Now, first of all, when we talk about this insecticide, not pesticide, insecticide. Because I was typing very fast, this one I put pesticide just now. Okay, insecticide will kill the larva and this will cause the mosquito population to decrease. Okay, so it's actually a very simple question, guys. Don't overthink it. Sometimes you all tend to overthink. Yeah, sometimes they just want a simple, direct answer. Okay, K-bar always got two types. The first type is they test you on whether you know the function. Example, a very simple one. If let's say a nucleus cannot function, what will happen? You know, that kind of very direct question. Another example is let's say um, the person lacks synovial fluid, what will happen to the joint? Yeah. So there'll be more friction, and this causes pain to the individual. So this is the first type. Second type is a bit more to common sense. This is second type. This is second type. Okay, next, Form 4, Chapter 9. Guys, Form 4, Chapter 9. Organ P is obviously small intestine. But if you want to write ileum, I think it's okay as well. You can write ileum as well, but small intestine is okay lah. R is labeled here. This is the appendix. Yeah, do you know I removed my appendix during the end of Form 4? I self-diagnosed. I self-diagnosed myself when I was Form 4. Long story short. Yeah. Okay. Those of my students know quite a funny story. I self-diagnosed. I went to the hospital myself. Then I realized I got no money to pay for surgery. I had to quickly call my dad to U-turn come back. Yeah, I'm very independent. But anyway, that's not the main point here. Look, Q is the colon. Colon is large intestine. Now, early stages is water reabsorption, but towards the end, because it is very near the rectum, Okay, you can put defecation. You can put defecation. B, discuss the bad effect for this long-term procedure. Actually, this is based on, I guess, a little bit from SPM 2024, because if you checked out SPM 2023, 23 or 24, there's this surgery to remove fat. 2023. 2023, question 11, there's this obesity surgery package, yeah, premium package. Now, the bad effect for this procedure is if you remove an organ, definitely less nutrients there'll be your digestion won't be efficient, you will lack nutrient, uh there'll be malnutrition, okay? May cause long-term diarrhea, and yeah, increased risk of small intestine complication. Now, the thing is this, whenever got surgery, there's also a risk of infection. There's also a risk of infection. And the last one here, you can just check. You can just check. Okay. Now, anyway, this diarrhea, right? The O should be here. Without the O is American. It's not wrong, lah. It's just American. Like photosynthesis, the S or Z. We follow S because we were colonized by the Brits before. We were colonized by the British before, so we had to follow them more. Now, if you forgot Pika, Pika is eating something that is not food. It is in your syllabus. I don't say, hey, sir, what is this Pikachu? No, no, no. It's in your Bug 9, Form 4 Bug 9 under disease section. Okay. I definitely have to go for counseling. They eat things that have no nutritional value, like these are shampoo, soil, paper, sheet. Yeah, she got no nutrient already. Okay. All right, guys. Question 8, Form 5, Chapter 11. Now, this Chapter 11, last year, I think, to be fair, not only myself, but quite a number of teachers spotted this topic because prior to 2024, sebelum 2024, I think 5 years they never asked. It was already high time. So good that it came out as a structure, lah. I think a complete structure, which is dihybrid. Dihybrid. Okay, guys, rhesus negative is always homozygous. Both must be Rh negative. Now, if positive, then since the child is Rh positive, okay, the negative is from the mom, and this is from the dad. Yeah, because the mom is negative, can only contribute negative. And they got mentioned this, sir, how do you know that this is positive and not negative? Because they mentioned father homozygous dominant. If you already know before I mentioned that, then good, you did read the question. But if you're wondering, sir, how come you just put there Rh positive, then I caught you already. You never read the question. Guys, you cannot just assume. You cannot just assume. Yeah. I'm very proud of my students who follow me for this chapter. I seldom say this in class also. Out of the 28 chapters, only this chapter I mentioned. I'm very proud of students who follow me for inheritance because when they go up Form 6, I really see a difference. I really see a difference. Pelajar yang ikut saya untuk pewarisan dan pelajar yang tidak. But it's not a problem lah. Because I'll take one class to do basic stuff. Okay, explain how the child shows the phenotype in 8A1. So, positive is dominant, negative is recessive. So, like, the sperm carried one Rh positive, and this, okay, it fertilizes the ovum that contains Rhesus negative allele. Okay. Right. So the zygote, the kid is Rh positive, Rh negative. Okay, guys, B1. Wow, this is handwritten. Nice. Oh, keguguran, not nice, not nice. So this shows the pregnancy history of a mother. Based on the table, predict what will happen to the fetus in the third pregnancy. Yeah, if the mother does not take any treatment. So if you realize, okay, ini anak pertama, ini anak kedua, keguguran, ya, keguguran. Then oh, oh, it's in BM and I thought got okay. So yeah, first kid is fine. First kid is fine. Second kid is not fine. Now, why is because of this? Look, look. The mom is negative, the child positive. I always say this in class. Our body will always attack something that it doesn't know. It's just like you. Will you ever just simply open your gate for a stranger? Hey, hello, I know your dad. We cycle to school. Yeah. Oh, wow, your cycle to school. I open for you now. Cannot. Guys, cannot. Same goes to your body. Defense means defend. Kalau saya ialah sel darah putih. If I'm a white blood cell, oh, I don't recognize you. I will attack you. Yeah. So that's how it works in our body. So same thing here. Because the mother's body does not recognize Rh positive. So if you remember what you learned in Form 4 Chapter 10, okay, the first child will be safe one because why? Concentration of antibody is low. Okay, let me move back to that slide. This one here. Sir, I don't understand. Since you say that the mom's body will attack the child, right? Then why did the first one survive? Guys, you have to remember your theory. Concentration of these anti-D antibodies for the very first is low. So the first baby is safe. Second baby is not safe because by then antibody concentration is high. And if the third one also no treatment is taken, the third fetus will die if he or she is Rh positive. Okay, personally, I find this question a bit lacking in information. They should state if the third child is Rhesus positive. If not, it's a bit too wide in terms of the scope. The scope. But it's still okay lah. It's still okay. I mean, I'm just being a bit particular. Suggest steps so they require a blood transfusion and the fetal have to be replaced with Rh negative blood. Okay, oops. Hold on. Okay, we need one more point here. We need one more point. Because it's 3 marks. So we can further elaborate the above sentence by saying that this is to ensure the mothers we can say defense or you can just say mothers anti-D antibodies in blood do not destroy the fetal red blood cells. Okay. In fact, she should go for there's this thing called anti-rhesus. Guys, there's this thing called anti-rhesus globulin treatment. Okay, should be taken. it should be taken especially after the first pregnancy itself. So this is to ensure that it won't happen. Okay, even if the child is positive, the mom negative. All right. So guys, that's for the structure questions. Okay, for this biology paper 2 set of Wilayah Persekutuan Kuala Lumpur. I'll see you for the essays next. Okay, in the next video. All right. Thank you.